All right. Hello, everyone, and welcome to your last exam review. So, oh, whoops. All right, so let's just jump right into it. Yeah. So basically, right, as you might have noticed, there's a lot of genetics talk in this exam, and that's for a reason. Dvorka has taught genetics in the past, right? So you've heard a lot of stuff about epigenetics, epigenomics, pharmacogenetics, that sort of thing, right? When ordinarily this would not be taught in this course, but in biology 302. So if you're a bio major, then you've gone ahead and had a bit of a head start with this class. In any case, I will be going over the practice exam today. I'm going to try and do it a little quicker than I normally do. And please bear with me. I'm not as familiar with this material as I am with some of the earlier stuff, but we're just going to go ahead and do it together. So, just jumping right into it. Um, for the true or false questions, the answer is just going to be there because I can't really help it. Okay, so true or false? Recessive mutations need to be paired with another recessive mutation in order to affect the phenotype. That is true. Now, what is a mutation? A mutation happens in the DNA, right? So if we have for example the mRNA that's being mutated, well that's just one mRNA that's being affected, right? One protein, maybe a few proteins are being affected by that one mRNA, but other than that, no big deal, right? However, once we have mutations in the DNA, then we start to get um, problematic, right? And there exists dominant and recessive mutations, right? In this case, we're talking recessive mutations. So if we have our chromosomes, right? If we have our chromosomes and we have a mutation here, if it's recessive, we need to have it on both genes, right? On both chromosomes for both alleles, right? So whether or not it's a beneficial or negative mutation, most likely negative, it will affect the phenotype if they're both recessive. Because remember, if we have just one dominant gene, in this example I'm using capital and lowercase d, if we have just one dominant gene, then that phenotype is expressed 9 times out of 10. I mean, in the case of like codominance or incomplete dominance, it's a little bit different. But in this case, recessive mutations need to be paired with another recessive mutation in order to affect the phenotype. That is true. So if we had this, this is going to affect the phenotype. All right, moving on. So number two is asking, maternal lack of care in rats can result in a long-term stressed phenotype in its offspring due to epigenetic modification, although this effect did not occur in utero. This is true. Whoops. This is true. So now why is this true? So this is like your number one or one of your number one examples of what it means for there to be an epigenetic modification. So what are epigenetics? Epi means above, right? So we're literally talking about above the genes. We're not talking about the DNA itself. We're talking about how the DNA is being affected by certain proteins like histones, certain functional groups like methyl groups or acetyl groups, or certain other certain things like microRNAs. So when we're talking about epigenetics, we're talking about, like I said, methylation, acetylation, microRNAs, right? So these things happen to the stuff around the DNA. So that's why it's epigenetics, right? It's above the DNA. So maternal lack of care in rats that could result in long-term stressed phenotype in its offspring. Well, I mean, I know it's a very common insult, like, oh, he or she didn't get enough love as a child, so they're very negative towards everything. That is actually true, right? And here's the evidence to prove it. I mean, when we're talking about epigenetics, I mean literally there's something psychological about a child rat being groomed by its mother, being properly groomed, and then a rat being abused by its mother. I mean literally it says here, right? Long-term stressed phenotype can be a result of lack of care, right? This is your number one example of how epigenetics affects the phenotype of an offspring, even though it did not occur in utero. It happened after it was born. So that's epigenetics. Like a, so that's why studying epigenomics is so important, right? Which is why Dvorak has a lab in it. Which is why you're probably going to get a few more questions about epigenetics on your exam. So I would pay a lot of attention to this. So that's true. Moving on, number three. All right, this is another question regarding epigenomics. Um, let's see. Epimutations can be passed via both female and male germlines. This is true, as you can see here. This is true. So what does this mean? So we talked about epigenomics, right? Epimutations, same thing, right? So if we have a sperm cell, excuse my very crude drawing of a sperm cell, this sperm cell, if you're a man, right, this sperm cell can be affected by your behaviors, right? So that includes like eating poorly or having poor health habits, that sort of thing. It could actually negatively affect the DNA in your sperm, right? And same thing for women. If you have the egg, right? I guess the egg's a lot bigger, right? A lot bigger. If we have the egg, same thing. If you eat poorly, if you have poor health habits, the DNA in here can be negatively affected, right? That's again, once again, that's epigenomics in action, right? This could be negatively affected. We have like a methyl group here, or we have like a acetyl group here, right? That and these can be passed down through generations, right? If you smoke during pregnancy, for example, that's happening in utero, you're affecting the DNA of the child. Or if you eat poorly, you're affecting the DNA of the sperm. That sort of thing, right? So mutations can be passed via both male and female germlines. That is true. Moving on. So number four, once again, another epigenetics question. So when I said that Dvorka works in a lab that focuses on epigenetics, I wasn't lying. There's a lot of epigenetics on this exam. So that should be your number one thing to focus on. I made the mistake of focusing on some of the Mendelian genetics. While Mendelian genetics is great, it's also very easy and very, I don't want to say easy, but it's very simple to understand. That's like high school biology stuff. So now we're focusing in and we're honing in on epigenetics, right? I mean, there's like there's two questions about Mendelian genetics. Anyway, number four is asking DNA methylation and transcriptional regulation via microRNAs can serve as a mechanism of epigenetic modification. Once again, true. So, what does this mean? DNA methylation, okay, that's epigenetics, right? Transcriptional regulation via microRNAs. Once again, epigenetics. And that's what the question is asking. These can both serve as mechanisms for epigenetics. So, what do microRNAs do? So microRNAs are tiny little sequences of RNA. I guess it's not really tiny, but smaller than your average, like tRNA or mRNA. What these will do is if we have like an actual fully fledged mRNA, so this has been transcribed, it's all ready to be translated. If we have the microRNAs, this can actually go on and affect how this is translated, right? So transcription regulation via microRNA. I guess this would be translation regulation. So microRNAs in this case can also affect how this forms, this mRNA forms. Not only how it's translated, but how it's transcribed. So microRNAs, um, I'm not sure if you guys know this, is it's actually a rather recent discovery, right? Within like the last 10 or 15 years, right? So genetics is a constantly evolving field. Just in this class we're talking about CRISPR-Cas9. So who knows, maybe in 10 years we'll have designer babies, which I don't know about you guys, but I'm not really a fan of that idea. It's kind of going to the dystopian sort of future pathway. In any case, we're talking about microRNAs. These serve as mechanisms of epigenetic modifications, and DNA methylation. That's like one of the number one. Once again, one of the number one examples of epigenetic modification: methylation, acetylation, and microRNA regulation. Moving on. So number five, I think number five was just given to you for free because this is kind of an unfair question. But here's how I answered it. Even if you don't know what a cDNA is or how recombinant DNA technology works, you can still answer this question pretty simply. Human insulin production via recombinant DNA technology requires the use of cDNA produced via reverse transcription from mRNA obtained from healthy humans. So I'm just now realizing that it's not as simple as it seems. Okay, I initially I said this was false. I mean, the question is false for one. Initially, I said it was false because mRNA from healthy humans. Well, we don't make insulin in healthy humans. We make it in prokaryotes, right? We make it in bacteria. However, we do need the mRNA from healthy humans. Okay, well, I don't know. Once again, not my exact field of expertise. Um, and so once okay, let's step back for a moment. We don't really need to know this, right? So why are we going over it? So let's just move on. Yeah, I would advise that you do know what recombinant DNA, just the basic technology of it is, recombinant DNA. However, this goes beyond the scope of this class, this question. So I wouldn't worry about it too much, and I'm not going to worry about it right now. So moving on. So once again, question six, epigenetics. That's like what, four out of the six we've gone over so far. That's a lot. So, number six, small non-coding RNAs. So what is that? That's microRNA, right? Can serve as an epigenetic vector, passing the effect from one generation to the next. That is true. Once again, true. So, what are our three major types of epigenetic factors? MicroRNAs, methylation, acetylation. So when we see small non-coding RNAs, well, that's just a microRNA, right? So epigenetic vector, well, what does a vector do? A vector leads something in a specific direction and power level, right? So microRNA, that's an epigenetic factor, right? That affects how the genes are expressed via epigenetics, right? And and epigenetic factors can be passed down from generation to generation, as we saw with the example of the germline epimutations, right? So the answer to this would be true. Moving on, number seven. All right. So once again, very recent discovery. Next generation sequencing using fluorescent labeling of dNTPs. So what does this mean? So next generation sequencing, right? That just has to do with like how we sequence DNA and how we track certain parts of the DNA, like our genes or our transcriptome. I'm gonna write that down actually. So that's how we keep track, right? Next generation sequencing. So fluorescent labels. So that just means what this just means is that we can keep track of where the inserted DNA was. So we could actually see how the DNA is being affected. Or yeah, dNTP stands for deoxy nucleotide triphosphate. Now, what in the hell does that mean? Well, you remember ATP, GTP, CTP? All of these are nucleotide triphosphates. Now we're talking about deoxynucleotide triphosphates. Essentially, these are just think of these as like your A's, your T's, your C's, and your G's. These are just nucleotides that are going to be added to the DNA. Don't even worry about it. DNTPs, those are nucleotides that are being added to the DNA. For fluorescent labels, these are DNTPs that we want to keep track of using next generation sequencing. So the answer is going to be true on that one. Moving on. Okay, I really don't like this question because I feel like it's intentionally misleading, and it's like kind of like a gotcha question. So, I guess it's spelled incorrectly too. Taq polymerase denatures at temperatures higher than 90 degrees Celsius. Now, this is going to be false. Why is it going to be false? That's because Taq polymerase denatures at a temperature of 95 degrees Celsius. Yeah, that's bullshit, isn't it? Like, how are you supposed to know specifically, okay, Taq polymerase denatures at 95 Celsius? You're not. That's just such a specific detail that you really shouldn't be expected to know. But you do, because it's fair game, right? 95 degrees Celsius. Just a quick Google search shows us this is true. But Taq polymerase denatures at temperature higher than 90 C, that is false. Now, what is a Taq polymerase, right? A Taq polymerase is basically what we use for PCR, polymerase chain reaction, right? And how we do that, right? So we basically just have a bunch of DNA, and what we want to do with PCR is we want to split apart this DNA, and we want to take the Taq polymerase and just make a ton more DNA. When I say chain reaction, I really mean chain reaction. Just think of like a nuclear reaction. You start off with 1, and then you go to 2, and then you go to 4, and then you go to 8, etc. Right? Same deal here. We're just making a ton of DNA so we could sequence it really easily. So that's PCR, right? And for PCR, we use Taq polymerase. Now, Taq polymerase has a very unique property in which it denatures at a very high temperature. 95 degrees Celsius, that's almost boiling temperature, right? Taq polymerase is able to survive that temperature. And the reason why we want it to survive such a high temperature is because DNA will actually start to split apart at higher temperatures because they're held together by hydrogen bonds, right? So when we break apart the hydrogen bonds, we're able to put in the Taq polymerase, and the Taq polymerase is able to read the DNA. It's able to make more DNA, right? It's able to make more DNA, and then scientists can go ahead. I guess I'm gonna draw like a pair of glasses. Scientists can go on and inspect this DNA and see, okay, well this is our sequence. That's basically what PCR is for. Polymerase chain reaction, Taq polymerase, use it to sequence DNA. So, sorry, there's a fly on my wall. I had to take care of it. Number 9, cancer is a polygenic disease since it involves multiple mutations and multiple genes. That is true. Remember, on Monday, we talked about cancer, right? What a fun topic, right? Who doesn't love cancer? I'm just kidding. Everyone hates cancer. Cancer is the worst thing ever. Isn't it not? Cancer is a polygenic disease. That is true. Why is that true? Because if one gene went wrong and that just gives us cancer, we would all be dead by now. Would that not be true? I mean, not all of us, obviously, but like there would be a good portion of the population that would be dying to cancer, right? If it was just one gene that needed to be involved. So these genes are known as proto-oncogenes. Onco meaning cancerous, right? Proto meaning first or before. So when we talk about proto-oncogenes, these genes, what they do is they actually code for, normally, they code for just cell cycles, cell regulation, that sort of thing. So when these genes go out of control, when they go crazy, then cell division starts to go crazy, and then we start to get tumorous growths, right? Which is cancer, right? So cancer would have to be a polygenic disease. Multiple of these proto-oncogenes need to be affected. And how does this affected? By carcinogens, by mutagens, by old age, by genetics, that sort of thing. So but ultimately it does have to involve multiple mutations at multiple genes. So that's why you shouldn't stay out in the sun too long, because the UV rays will eventually break down your DNA, causing mutations. So, wear sunscreen. Moving on. Question 10. All right, question 10. RNA sequencing allows us to interfere mRNA sequence via sequencing of cDNA. Wow, what does that even mean, right? Okay, RNA sequencing. I'm gonna read it again. RNA sequencing allows us to interfere mRNA sequencing via sequencing of cDNA. So this one, it says it's true. Let's take its word for it. This is true. So cDNA is complementary DNA, right? Did I say interfere? It says infer. Sorry. One sec. I just don't. I want to make sure that I'm not saying anything wrong. Okay, so this is RNA sequencing. Yeah. Okay. So cDNA. We went over in question 5. We kind of skipped over question 5 because that question was not very good. What is cDNA? It's cDNA is complementary DNA. So basically, right, what we want to do, so for one, for starters, we're gonna do this. So why are we doing this? I need to slow down a little bit. Sorry. Why are we doing this? Well, we want to figure out our transcriptome. We want to figure out how our DNA is being actually like transcribed and how it's being used, because there's a ton of DNA in your cells that just straight up isn't used. It's just kind of like it's not used for anything. I mean, we only have like 20% of actually coding genes, so we want to see what these genes are through our transcriptome that are being, uh, you know. Anyway, so cDNA, what this will do, right? So cDNA is complementary DNA. So if we have an RNA, if we have an mRNA, right, what we could actually do is we could predict what the sequence we could predict the sequence in which it originated from via cDNAs. So these cDNAs will actually track down the RNAs and it'll bind to it. So we could actually figure out where this originated from. So RNA sequencing allows us to infer mRNA sequence via the sequencing of cDNA. That is true. I'm going to say it again for my own sake because this is a question that I'm, again, not so familiar with. RNA sequencing allows us to infer the mRNA sequence via the sequencing of cDNA. So the cDNA is something that we know. We know exactly what the cDNA consists of. What we want to know is the mRNA, and that's why we do RNA sequencing. Moving on, because I'm sure you're tired of hearing me talk about sequencing and all that jazz. So, um, where is okay? Here we go. So we went over one question that regarded like Mendelian genetics. Here's your other question. A true breeding green-skinned frog is mated with a blue-skinned frog to create an F1 generation. The F1 are then mated to produce the F2 generation. Upon close examination of the green-skinned frogs in the F2, 3/4 had stripes, 1/4 were unstriped. So, at this locus, at this genotype locus of the S gene, what was the genotype? So here we're just doing a basic monohybrid cross, right? I think the question was a little bit confused, or I think Dvork forgot to change up some of the yeah. Anyway, green skin frogs and blue skin frogs, and then we're talking about later whether or not they have stripes. We're just gonna assume that green skin frogs actually means stripes, and unstripes means blue skinned. So we're just gonna assume that, right? So if we have a true breeding green skin frog, that means we have what is it? S. We have capital S, and then blue skin frog, which we're gonna assume is recessive. So if we cross these, we have one gamete combination here, one gamete combination here. Our only possible outcome is this, right? So if we cross this, right, if we cross this with itself, we will then get the following possible combinations. 25% here, 50% here, and then 25% here. So 25% of their offspring will have no stripes, or will be blue skin, depending on how you see. Okay, so working backwards, right? So 75%, which that's 3/4, right? 75% of the F2 generation will have green skin, right? 75% will have green skin, and then 25% will have blue skin, right? Or will be unstriped. Again, it's confusing. Let's just focus on the color of their skin and not whether or not they have stripes. 75% do have stripes. Sorry, 75%. See, I'm confusing myself now. Okay. So anyway, if we work backwards, we could see, okay, well, this follows the 3-to-1 ratio that we talked about, right? So if we have this cross, right? If we always have this cross, whether it be this or this, if we always have a capital with a lowercase and then a capital with lowercase, that will always result in a 3-to-1 ratio, assuming there's not any other sort of factors at play. That's always going to be a 3-to-1 ratio. And what is it here? 3-to-1 ratio. So we could then assume that the F1 frogs are going to have this genotype, B. It can't be C and it can't be A because that would result in a different ratio. It has to be B. So moving on. So yeah, here's a good one. Agents of epigenetic modification is or are DNA methyltransferase, histone acetylase, microRNAs, all the above or none of the above. What did we talk about? What are our three primary epigenetic factors? DNA methylation, histone acetylation, and microRNAs. Well, what do we have here? DNA methyltransferase, which transfers a methyl group onto the DNA. Histone acetylase, which acetylates the histones, right? The ACE, it means it's an enzyme, right? And microRNAs. That's all three. So that's all the above. Just it's really important to remember what these do, right? So the microRNA will actually affect transcription translation. It will actually affect the rate of which it happens. Histone acetylase, if we have more acetyl groups, there's going to be more gene expression. If we have less acetyl groups, there's going to be less gene expression because acetyl groups, what those do on the histone, what the acetyl groups do on the histone is that they actually loosen up the DNA, making them available for transcription. And DNA methyltransferase does kind of the opposite. Well, it doesn't do the opposite. What it does do, however, is that it adds a sort of like flag to the DNA, and this flag, let's see if we have our DNA here. So if this is our DNA, that's really really bad. Sorry. If we have a this is our methyl group, for example, and then we have the polymerase coming this way. Well, once we hit this point, well, it doesn't want it can't go anywhere. It can't go any further. It acts as like a flag. It acts as a gene silencer. That is what a DNA methyl group does. So the answer is D, all the above. Moving on. Number 13. So, number 13 is asking epigenetic changes are caused by environment, yet can be inheritable from one generation to the next. Because A, germline cells develop epigenetic changes and carry them to the next generation. B, somatic cells do not develop epigenetic changes. Or C, changes in DNA sequence are corrected by repair machinery. Okay, so for your exam, I'm not sure if you're going to get 3 answer options or 4, but 3, well, 1 out of 3 chances is pretty good to me. Somatic cells do not develop epigenetic changes. Well, we know that one's wrong because of the rat example earlier, right? That one can't be right. So, C is saying changes in DNA sequence are corrected by repair machinery. Okay, well, we're not talking about the DNA sequence, are we? We're talking about epigenetics. We're talking about above the DNA sequence. So C is going to be wrong, leaving only A. Germline cells develop epigenetic changes and carry them to the next generation. Once again, smoking pregnant lady. That's going to do really bad things to that child in there. It's going to cause epigenetic changes in utero. It's going to cause, and if, going back to the example of the male with the bad sperm, I mean, the male with the bad sperm, he eats like crap, and he smokes and he drinks, and his sperm are very they're very epigenetically changed because of that, for that reason. So they can be passed on from generation to generation. Moving on. Okay, number 14 is asking. In recombinant DNA methods, the term vector can refer to A, the enzyme endonuclease that cuts DNA into restriction fragments, B, the sticky end of a DNA fragment, C, and circular endogenous DNA in mammalian cells, D, a plasmid used to transfer DNA into a living cell, or E, a DNA probe used to identify a particular gene. So, again, what is a vector? A vector, just in physics or in chemistry or in anything, what a vector does is that it shows us the direction and the magnitude at which something is being directed, right? So that's what a vector does. So a DNA probe used to identify a particular gene, well, it can't be that because we're not actually changing anything. We're not transferring anything, right? It can't be B, the sticky end of a DNA fragment, because once again, not transferring anything. Sticky ends are sticky ends. They're not vectors. A, the enzyme endonuclease that cuts the DNA into restriction fragments. Once again, not a vector, not transferring anything, not moving anything. So that leaves us with C or D. C is saying a circular endogenous DNA in a mammalian cell. Well, I'll say this. What are mammalian cells? Eukaryotic cells. What do eukaryotic cells not have? Circular DNA. We can eliminate C, therefore, leaving only D, a plasmid used to transfer DNA into a living cell. Well, that checks all the boxes, doesn't it? A plasmid, right? So a plasmid is a tiny little bit of DNA outside. So if we have like the nucleoid region, let's say this is a nucleoid region, we have the big old circular DNA. And we have the bacteria, right? We have these plasmids that go out, and these plasmids, what they do is they affect transcription. They add genes into living things, and those act as vectors. Therefore, recombinant DNA. That's what this is for. These vectors are going to go out and they're going to transfer DNA into a living cell. That's what these plasmids are for. They exist outside of the nucleoid in the prokaryotic cell. So, moving on. Number 15 is referring to palindromics, I guess you could say. Which of the following sequence could be a restriction enzyme recognition site? So, remember, what do we want to do with the restriction enzyme? What we want to do is we want to go into a specific sequence of DNA and we want to cut it at that point so we could go ahead and insert it. So in this case, right, if we have A, T, T, I'm just going to say it now. The answer is A, just to simplify it. A, T. Okay, well, what's that backwards? All right, now this is something that an enzyme can go ahead and recognize. So whatever we have on the outside doesn't really matter, right? Who cares what we have on the outside? What we do care about is this region right here. So let's do that in orange, yeah? We care about this region because this region is considered palindromic, right? ATTAT, ATTAAT, ATTAAT. It's the same forwards as it is backwards. So B would not be an example of that, and C would not be an example of that because they're not the same forwards and backwards. So let's just sequence it out real quick. Yeah, would not be the same thing, right? It would be slightly different. This would not be the same thing either. These would both be different. So this is the most common type of palindromic site, ATTAAT. So what happens is that the enzyme will come in and it'll split apart this DNA kind of like this, right? So once we do this, right? Let me see if I could do this. All right. So, all right. Once we do this, oh yeah, that worked perfectly. Now we have what's known as sticky ends. So now these sorry. Now what these sticky ends can do is that we're actually going to go ahead and insert new DNA. So now this DNA is new. This is a new gene that's able to be transcribed and be made into a protein. That is the basics of recombinant genes. We're splitting apart a palindromic site. We're inserting something that we do want. So hence we're recombining it. Right? We're splitting it apart, putting something in new, and we're combining it again. And what happens with these little sites? Well, a little enzyme known as a ligase comes in, and it actually fixes it together. So we would actually have a more uniform DNA sequence. So it's not just hanging on by a thread. We actually have the glue sticking it together. We have a covalent bond sticking it together via our ligase, and that's ligase spelt ILIGASE, ligase. So that's the basics of recombinant DNA. It all starts with one of these, a palindromic site. Moving on, number 16. So once again, this is talking about cDNA and what that means. So number 16 is saying blank is required to make a single-stranded cDNA from an RNA transcript. So the answer options are Taq polymerase, helicase, reverse transcriptase, or primase. Okay, so a lot of you, like a good portion of you, don't remember DNA replication, and that's totally fine because I don't think we really went into it in a whole lot of detail. Helicase and primase are two enzymes that we worry about in DNA replication. Well, here we're not worrying about DNA replication. We're talking about making a single-stranded cDNA from an RNA transcript, right? So how do we normally get an RNA transcript? We make it from DNA. So if we do it in the reverse, so normally we go DNA to RNA, right? Well, what happens if we go from RNA to DNA? Well, this would be a inverse process, right? Do you see what I'm getting at? Reverse transcriptase. ACE meaning enzyme. Reverse transcription referring to the fact that we're literally going backwards from the RNA to the DNA. Reverse transcription. That's what this is doing. And it's required to make a single stranded cDNA, complementary DNA, from an RNA transcript, an mRNA, right? So we need reverse transcriptase to actually do this, to actually make the cDNA, to actually be able to see our transcriptome, to see what's able to be transcribed and what we're actually making in our DNA. Do you see what I'm getting at here? All of this connects together, right? Moving on, number 17. We're halfway through, I think. PCR. This one's talking about PCR. We were just talking about PCR, right? So, in PCR, DNA replication is carried outside of the cell in a tube. The only enzyme provided is Taq polymerase. What performs the function of the remaining enzymes that are used during replication, such as helicase? So, once again, you guys didn't really go over DNA replication in a whole lot of detail. So if you don't know what a helicase is, well, you're kind of SOL, right? So what a helicase does is, so if we have our DNA, so I think I could find an actual like picture of this so you could see what I'm talking about. This is going to be a PNG, so yeah. So this is our DNA, right? Right here and right here. So this is DNA being formed, right? Over here, this little blue enzyme here, that's our helicase. So we have to actually split apart the DNA before we do anything to it, before we make more DNA. In this case we're just making more. We're duplicating DNA for it to be we're duplicating DNA so we could then go on to split apart during metaphase and everyone's happy, you have the proper number of chromosomes, yada yada yada. We need to actually split it apart first. And that is the function of the helicase, right? So I'll write that down. I'll even write down in blue. This is the helicase, right? So, what performs the function of the remaining enzymes, such as helicase? So the Taq polymerase is actually doing the heavy lifting, right? It's making more DNA. So we could go ahead and eliminate Taq polymerase because why would we use Taq polymerase to perform the function of the remaining enzymes? Taq polymerase only does one thing, and that's make more DNA. Magnesium ions, we never talked about magnesium ions, so don't even worry about it. The buffer solution is just the solution that exists during PCR. It's to keep the DNA properly. It's to keep the DNA nutrition proper and to keep it in a sort of buffer, so that sort of thing. But it doesn't perform the actual function of the helicase. What does perform the function of the helicase is high temperature. Because remember, what holds together DNA? Hydrogen bonds. How are hydrogen bonds broken? High enough temperatures, right? So if we bump up the temperature, well, we're actually going to perform the function of the helicase via high temperature, leaving D the correct answer. Because remember, Taq polymerase has an incredibly high it functions at an incredibly high temperature without being denatured, right? So, CRISPRs are naturally found in A, bacteria, humans, yeast, or plants. So, this one's, if you don't know it, it's just kind of one of those things where it's like if you know it, you know it. If you don't, well, you're kind of out of luck. We do not make CRISPRs. Humans do not make CRISPRs. Eukaryotic cells don't make CRISPRs. So it leaves bacteria and yeast is a type of bacteria. Well, if CRISPRs were naturally found in yeast, well, then CRISPR would probably be discovered a lot earlier than it was, and it would probably be a lot cheaper than it currently is as well. So it's just, like I said, it's just kind of one of those things that you have to know. CRISPRs, I can't remember the acronym. The acronym doesn't really mean anything. I just remember palindromic something. CRISPRs are a defense mechanism for bacteria cells, right? So CRISPR's, if there's an invading bacteria, for example, it could just CRISPR can go in and cut up the DNA and then make it ineffectual. So we have gone and used this for our own benefit, just like humans always do, right? So the answer is going to be A. It's not going to be yeast. Yeast would be a good guess, but if it were yeast, then it would be a whole hell of a lot cheaper. It's just general as a general idea. It's found in bacteria. Now that's how many species of bacteria there are. Probably like billions. So just choose like a really general option for this one. Moving on. Question 19 is asking the following. Okay. So, after a fragment of DNA containing the gene of interest has been inserted to a vector, how are the gaps between two pieces of DNA sealed together? A. DNA polymerase catalyzes the formation of hydrogen bonds in the DNA backbone. Well, for one, we could go ahead and eliminate that because there aren't any hydrogen bonds in the backbone. B. Reverse transcriptase catalyzes the formation of covalent bonds in the DNA backbone. Once again, that would not that we're talking about two different things here. Currently, we're talking about um. So B is describing how to find the transcriptome, right? It's describing how we would use how we would find how the mRNAs are expressed. The question is asking about recombinant genes, so we can go ahead and eliminate B. C, DNA helicase causes the double helix to wind into its stable form. Well, that's wrong because we just discussed what a helicase does. It does the exact opposite. It unwinds it, right? And D is saying restriction enzymes have the ability to join together two DNA fragments. Well, we talked about a restriction enzyme earlier, didn't we? With the sticky ends, right? With like the with uh I don't have it now, but we talked about like the A with a palindromic sequence, right? We talked about that how we would cover it here. Trans restriction sorry restriction enzymes have the exact opposite effect. They don't have the ability to join together two DNA fragments. They actually split apart a DNA fragment so something can be inserted. So the answer would not be D, leaving only DNA ligase catalyzes the formation of covalent bonds in the DNA backbone. That's what a ligase does, right? That one's correct. That's correct. DNA ligase catalyzes the formation of covalent bonds in the DNA backbone. That's exactly what's happening. Remember, that's what a ligase does. Let's see if I could find. Let's see, ligase. Okay, well this is even better actually, because this actually pertains to what we're talking about. So normally when we're talking about a ligase, we're talking about DNA replication. Here we actually do have the example of the sticky ends, right? These are sticky ends. These are palindromic sequences. These were split apart by a restriction enzyme, and now they're being put back together by a ligase, right? What's not pictured here is the actual ligase itself, but notice here how we have the actual bridges, how they're actually bridged together now. Because normally they wouldn't be bridged together like that, right? That couldn't happen. So here they're actually bridged together, and what does that? A DNA ligase. Moving on. Number 20 is asking about the reverse transcriptase again. Do you see a bit of a trend here where we're talking a lot about reverse transcriptase? We're talking a lot about epigenetics. This practice exam is a very good way of being able to know what's going to be on your actual exam. So please do yourself a favor. Do not study up on monohybrid, dihybrid crosses. Do not study up on co-dominance, incomplete dominance. Do not study up on Mendelian genetics. Study up on the hard stuff because the hard stuff is most likely what you're going to be tested on. That is my number one advice to you. Moving on. So, number 20 is asking what is the function of reverse transcriptase in retroviruses? So, what is a retrovirus? A retrovirus is a virus that works in reverse, right? Or it works backwards how it normally would. So, just just I'm just gonna say it. The answer is B, right? It would have to use viral DNA as a template for DNA synthesis. So what's a common retrovirus we talked about? HIV, right? So what HIV literally does is that it changes your DNA. You get HIV transmitted through blood, and then the virus will go in and actually change your DNA. It will insert itself. It will use the viral RNA as a template, right, for DNA synthesis, right? So it would. So that would that's what would sorry, that's what a reverse transcriptase would do in this case. It would not hydrolyze the host cell DNA because that would just kill the cell, right? If we're hydrolyzing the DNA, we're just cutting it up a bunch, right? It converts this host cell RNA into viral DNA. Well, that doesn't really make sense because that well it kind of does make sense. But okay, well here's what we want to do, right? If we're converting host cell RNA, well, we're just going to convert RNA back into the DNA, back into the host DNA, which is not what the retrovirus is doing. It's converting its own RNA into the viral DNA. Translates viral RNA into proteins. Well, it can't do that if it doesn't. It can only do it. It can do it very inefficiently. But if we added DNA into the equation, then it does it for the entire cell, right? And then uses viral DNA as a template for making complementary RNA strands. So once again, we're talking about reverse transcriptase, right? So why would we make complementary RNA strands from RNA? That's not what transcription is, so we can go ahead and cross that one out too, leaving only B. It uses viral DNA, viral RNA as a template for DNA synthesis. So the virus will make its own RNA. The virus has its own RNA, right? And it uses that. It inserts that as a cDNA into our cells. So if we're literally changing the DNA, right, you can see how that's really bad. You can see why an autoimmunodeficiency syndrome is really dangerous and was a death sentence for quite a few people a couple of decades ago. But fortunately, advancements in science, it's got a lot better. Anyway, sorry. So this question is asking about SNPs, SNPs, which stands for single nucleotide something. I honestly don't know. Single nucleotide polymorphism, right? So what that essentially is saying, do you guys remember your mutations that we talked about? Like point shift mutations and then single point mutations. That's essentially what we're talking about here. Here we're talking about single point mutations, which are the most, I would say, are the most common mutations that we're talking about. Single point mutations, right? How we would find those is if we have a large, database of quote unquote normal or healthy control variables, and we have that to compare it to someone who does have the mutation, right? And we would compare it to that, and we would look for these single nucleotide polymorphisms. We would look for these mutations in a single point and point it out and say that's where it went wrong. We have a bunch of people who are healthy who don't have this, and we have one person who has a single nucleotide polymorphism. This person has a genetic disorder or a mutation. At this point, case closed. So what I just described would be option B. Single nucleotide polymorphisms, where one allele is found more often in persons with a particular disorder than in healthy controls, right? What does C say, for example? Okay, C is saying single nucleotide polymorphisms where one allele is found in families with a particular intron sequence. Okay, well, introns are not what we care about, right? So just right off the bat, we could eliminate that, right? Single nucleotide polymorphisms where one allele is found in two or more adjacent genes. Well, that doesn't make sense because there's one allele for one gene, right? So there couldn't be in more. We couldn't find that in more genes, right? And then EU is saying large inversions that displaces the centromere. Well, that also wouldn't really make sense because one nucleotide is not going to completely displace the centromere. One nucleotide, right? It wouldn't be a large inversion. It'd be a very, very subtle and very, very small change in the DNA. And then A is saying lengthy sequences that might be shared by most members of the population. Well, once again, in large scale, genome-wide association studies in humans, we're not looking for lengthy sequences that might be shared by most of the population, right? We're looking for the small bits, right? We're looking for single nucleotide polymorphisms that change from person to person, or they don't change from person to person, that might change from person to person, and we can compare that to the population, which is healthy, right? So here, when we're looking at genome-wide studies, we're looking for the small bits, right? We're looking for the stuff that we can actually help people in. It does. I mean, it doesn't help us that we know. Okay, some people have brown hair, some people have blonde hair. Okay, hututu. Yeah, we don't care about that stuff. We care about the details, right? Sorry. Okay. So this is one of your slider questions. Blank is useful for useful in reducing adverse drug reactions. Epigenomics, pharmacogenomics, proteomics, transcriptomics. Okay. What are drugs? So what do you go to Walgreens for? You go to pick up a prescription, or you go to Albertsons or Walmart or CVS, wherever you get your medicine from. What is it called? A pharmacy, right? What is the only answer option with farm in its name? Pharmacogenomics. So what is pharmacogenomics, right? This is our answer, by the way. Pharmacogenomics is a field of study in which we discover how our genome affects how we break down or metabolize drugs. So pharmacogenomics would be incredibly useful in reducing adverse drug reactions for the following reason. Okay, let me give you an example. Last semester, I did a project on coding, and in fact, you could actually find that video on my YouTube channel. I did a project on coding, and coding is a really inefficient medication because some people metabolize codeine really, really well. Some people metabolize it really, really poorly. So what do you use codeine for? Codeine's it's a steroidal um, it's a steroidal painkiller, right? So some people metabolize it really, really well. It's too effective, and people can have adverse reactions from that. Or, on the other hand, if some people don't metabolize it very well, well then they're not getting the proper painkiller effect, right? So pharmacogenomics can actually be an effective way of reducing these adverse drug reactions. We can actually go ahead and see this person is safe to take codeine, this person is not safe to take codeine, or whatever drug, for example. It would not be proteomics, it would not be transcriptomics, it would not be epigenetics, pharmacogenomics. If you could hear a cat in the background, that is my little boy Link, who is quite loud. So you'll have to bear with me. So number 23, we talked about proto-oncogenes before, right? And he's currently on my desk. Sorry, give me one sec. Number 23, proto-oncogenes. We talked about those, right? What do? Proto-oncogenes are involved in the process of cell division and cell growth. And if we have cell growth out of control, if we have cell division out of control, then that's cancer, right? Onco means cancer, proto means before, so before cancer genes, right? So just think that through, right? What did I just describe? Right? I just described answer option D. Proto-oncogenes stimulate normal cell growth and division. So that's proto-oncogenes. That's before they're actually activated. Oncogenes would code for something that the oncogenes would code for increased cell growth and division, which leads to tumors, right? So proto-oncogenes are underexpressed in cancer cells. Well, they wouldn't be underexpressed in cancer cells. They just wouldn't be expressed at all because it would just be oncogenes at that point. So it can't be E. C are produced by somatic mutations induced by carcinogenic substances. Well, once again, that would be oncogenes. Here we're talking about proto-oncogenes before they're actually been activated into before they've actually been mutated into something that's dangerous, right? And then we have B, were initially introduced into eukaryotic cells by retroviruses. Once again, proto-oncogenes are normal. They're not. They're not foreign to us. There's something that's actually in our DNA that codes for normal cell division and growth. And proto-oncogenes normally suppress tumor growth. Well, that can't be the case. It's just out of simple definition, because this is the case, D is the case. Proto-oncogenes normally suppress tumor growth. I mean, that would make sense because if it then activates, right, then it wouldn't suppress tumor growth. That would make sense. But there are other genes that control for tumor growth, and it's not proto-oncogenes. It's proto-oncogenes perform D. So we have a couple questions left. This one's a bit of a long one. It has only 2 answer options. Okay, I'm gonna I guess abridge it a little bit for you guys because it is quite a bit. Or I don't know. Anyway, I'll just read it out. The BRCA1, BRCA2 genes are tumor suppressor genes. They're known as the breast cancer genes. People who have a mutation in one of these genes are at increased risk for developing breast cancer. However, not everyone who inherits a copy of mutated BRCA develops breast cancer. The nature of tumor suppressor genes is recessive and is often described by a, quote, two-hit hypothesis. Pick out the correct option that explains why an individual with just one mutated copy, might not develop cancer. And our answer options are the functional copy in an individual heterozygous 4 mutated tumor suppressor gene brings about the tumor suppressor effect and prevents cancer. Sorry. And B, the mutated copy of the tumor suppressor gene is inefficient in protecting against unregulated cell division. So I think what the question is basically saying, two-hit hypothesis. So this, so mm, so if I had to guess, this would be a case of incomplete dominance because. Okay, sorry. This is the question that is this is a question that's not ordinarily taught in this class. So basically, I think what the question is saying, how so if we have so normally people have this genotype, and this genotype codes for tumor suppression, right? So if we're both recessive, then then the cell's working properly. It's properly suppressing the tumors in breasts, right? If this mutates to a capital B, then still there's still going to be some tumor suppression because of incomplete dominance. There'll still be some tumor suppression here, but it's not going to be as effective due to there being a dominant gene here. And if we have 2, well, this means this means that it's totally unchecked. The tumor suppressor gene is not functioning properly, hence why it's called a two-hit hypothesis. So from this, we can then say that the mutated copy of the tumor suppressor gene is inefficient in protecting against unregulated cell division. We can say this one's actually wrong. And we can say that the functional copy, being, I guess, hold on, the functional copy being this copy, right? The functional copy in a heterozygous individual brings about the tumor suppressor effect and prevents cancer. So that is to say that people still have a increased risk for developing breast cancer in one of these genes. They have an increased risk, but they're not guaranteed to get breast cancer because this gene is still functioning properly as a case of incomplete dominance. Incomplete dominance basically means that the recessive allele and the dominant allele are both expressed. They're kind of in conflict with each other because the dominant gene isn't completely dominant over the recessive. So the answer will be A. That one was long. Let's see what time we're running on. Okay, we're just a little over an hour, so this is actually a little shorter than I normally go, which is great because I feel like I spend a lot of time on these sometimes. So number 25, and then we're gonna do a lightning round on the true or false questions. So this is once again talking about proto-oncogenes. Proto-oncogenes can change into oncogenes that cause cancer. Okay, we knew that, right? Which of the following best explains the presence of these potential time bombs in eukaryotic cells? All right, well, proto-oncogenes normally just help regulate cell division. Proto-oncogenes first arose from viral infections. C, proto-oncogenes are genetic junk. D, proto-oncogenes are mutant versions of normal genes. Or E, cells produce proto-oncogenes as they age. Once again, proto-oncogenes can change into oncogenes that cause cancer, right? So if they change into that, right, they wouldn't produce proto-oncogenes as they age. If anything, would produce oncogenes as they age, right? And they're certainly not genetic junk because they actually code for cell division and cell growth, right? And they wouldn't first arise from viral infections because that wouldn't really make much sense. Because once again, it's not it's not necessarily a bad thing that we have proto-oncogenes. They're very necessary for proper cell growth and division. And then we have D, proto-oncogenes are mutant versions of normal genes. Well, that's just not wrong that's not right because oncogenes are the mutant versions. So the answer is B. Proto-oncogenes normally help regulate cell division. There's been like 3 questions surrounding that one answer, right? So that's all you gotta know. Proto-oncogenes are normal. Proto-oncogenes are essential to a cell, and once those go out of hand, then they cause cancer. That's the one thing I want to emphasize. So, now, extra credit lightning round. Let's go. So, alleles segregate during gametogenesis. That is true. Diploids receive one copy of an allele from a gene from each parent. That is most definitely true. We learned that when we're talking about cell division and we're talking about homologous chromosomes. Diploids receive one copy, right? We have one from mom, one from dad, right? That's why you would most likely look like a mixture of your mom and your dad. Number 28, epigenome determines genetic expression. That is true. Don't let it fool you. That is true. Your epigenome definitely determines gene expression. If a mother has a child in her womb and she smokes and that affects the epigenome of that child is going to grow up with growth deficiencies. It's going to be really bad for the development of the child. The epigenome determines gene expression. That's what it does. I mean, DNA methylation. I mean, gene silencing, right? DNA acetylation or histone acetylation. Gene that increases the expression of the gene and microRNAs. All of that has to do with epigenetics, and all of that determines gene expression. That all has to do with gene expression. Once again, I'll reiterate what Dvorka said in class today. If you get that one wrong, we have failed you, because that has been made very clear. Epigenome determines gene expression. Number 29, reverse transcription PCR, reverse transcription polymerase chain reaction directly sequences RNA molecules by complementary pairing with microRNAs. That is false. There's a complementary pairing with cDNAs, not microRNAs, right? So reverse transcription, always remember we're going from RNA to DNA. When normally it would be DNA to RNA, right? But since we're going in reverse, it is the reverse of that. Number 30, comparative transcriptomics examines the difference in DNA sequences between different tissues. I guess that one's false. I really haven't heard of that one. That one's I'm afraid that's one's you're just gonna have to research on your own because I'm getting quite tired here. That one's just false. Comparative transcriptomics, I would look into that a little bit. I don't think it's gonna be super present on your exam. That one's false. I kind of skipped over number 26, so let's go over that one too. Number 26 is saying allele segregate during gametogenesis. Well, that one's 100% true. Remember again from meiosis, when we talked about meiosis. During metaphase 1, we're separating homologous chromosomes. Hence, we are separating alleles, right? We're separating the actual genes you get from mom and get from dad, and then it's going to combine again later with a different gamete as a sex cell, and it's going to create a whole new organism, right? Alleles segregate during gametogenesis. That is metaphase 1. That is from last unit, right? Just don't forget it. In any case, we are done. That was your practice exam. We're running about an hour and 15. This is taking a lot less long than I anticipated, which I'm really thankful for because I have my own work to do. Which, if you're hearing that, don't feel bad. This is my choice to do because I know you guys really like these videos. I know you guys really, I know you guys really get a lot of information from these videos, which I'm super thankful for. Throughout the semester, I've gotten a lot of comments about how these videos have been helpful to you guys, and I just want to say thank you for your input. That really does help. I'll do more of these in the future for the other PLF classes, but in any case, this is the last one that you guys will see. This is your last exam. You guys after Friday, you guys will be done with this class. Isn't that exciting? For some, this may be your first semester at UNM. For some, this may be your last semester. I don't know. Probably not because this is mostly a class of freshmen. And for many of you, it will be your second semester here at UNM. I just want to say thank you guys for a great semester. It's been really great. I haven't had a lot of you go into my office hours, which is okay, right? But if you have any friends that are going into genetics, sorry, if you have any friends that are going into 2101 next semester, please let them know that I will be there as well, and I will be doing these videos, and I will be hosting office hours. Please let them know because guys, I'm here to help you. I'm here to help you, and please don't be afraid to ask and reach out and ask any questions. With that being said, I wish you all the best of luck. I wish you all a good exam. I wish you all a happy summer. All of that jazz. I will see you all later. Bye.